we present the ultimate Bram-Cor utilities designed for a significant achievement in modern clinical research. TPN, Total Parenteral Nutrition. By delivering carbohydrates, amino acids, lipids, electrolytes, vitamins and trace elements directly into the bloodstream, TPN sustains metabolic functions and improves clinical outcomes in critically ill patients. We are at the Bram Cor workshop, where technicians are completing final calibration on an advanced TPN system, typically based on 3 sterile macronutrient streams: glucose, amino acid solutions, and lipid emulsions. As part of the broad family of large volume parenterals, TPN solutions are among the most demanding sterile medicinal preparations. That precision and formulation range is exactly what opens up promising market niches and real business opportunities. In Bram Cor TPN Systems, the principles of quality by design are proactively incorporated into every construction detail. Each stream maintains predefined critical quality attributes throughout the entire life cycle of the product, from formulation to final filling and storage. Clearly designed to operate under recipe-driven batch execution, all process steps are controlled by validated automation systems in accordance with predefined electronic batch recipes. Thanks to the modular software that runs as a browser, the entire system is manageable from any device, with no location constraints. This is an extraordinary comprehensive overview. A single interface to monitor every process, every parameter. Every machine is connected, recorded, and tracked in full GMP compliance. With all machinery connected, the TPN plant communicates as a unified organism. And now we present the first line to produce glucose solutions, the primary energy source. It all begins with the arrival of purified water or water for injection. The solvents, which travel through the storage tank and its loop, supplying the formulation dissolvers and mixing tanks. Here we're at the heart of the glucose line, featuring Bram Cor's engineered vessels and a sophisticated, precisely structured filtration group. Providing sufficient glucose reduces endogenous protein breakdown by limiting gluconeogenesis from skeletal muscle amino acids. This protein-sparing effect is one of the primary objectives of TPN therapy. The general process flow involves formulation, conditioning, filler buffering, and 3-chamber filling. The entire manufacturing process runs on recipe-driven batch execution, ensuring full standardization and traceability. A key design principle of the platform is the establishment of a controlled aseptic boundary, encompassing formulation vessels, transfer lines, filtration systems, buffer tanks, and filling machines. We're now at the loop and storage tank of the second line, producing amino acid solutions. In this line, water quality plays a particularly important role, because impurities or microbial contaminants may accelerate degradation reactions. That's why the only feed water used is compendial water for injection. Unlike carbohydrates and lipids, amino acids primarily serve structural rather than energetic functions. They provide the nitrogen necessary for protein synthesis, tissue regeneration, enzyme production, hormone synthesis, and immune function. As with the previous stream, the general process flow includes formulation, conditioning, filler buffering, and 3-chamber filling. The aseptic boundary here follows the same approach. Closed connections on aseptic valves and hygienic fittings. Clean in place and sterilization in place cycles, sterile filtration barriers where applicable, controlled pressure cascades, and automated valve sequencing. Now we present what sets Bram-Cor's TPN offer apart: the most advanced process flow to produce lipid emulsions. While the glucose and amino acid lines are consolidated by world standards, the ability to offer highly evolved, specialized emulsions represents the most exciting frontier in marketing three-chamber bags. Lipids are naturally insoluble in water. To enable safe intravenous administration, triglycerides are dispersed as microscopic oil droplets suspended in water, forming an oil-in-water emulsion. A dedicated homogenization stage ensures strict control over droplet size distribution, emulsion stability, physical integrity of the formulation, and compliance with parenteral emulsion specifications. Advanced process control, fully open formulation, precision in the homogenization stage, high-quality filling with buffering. These are the reasons behind Bram-Cor's superior design, and every step remains fully traceable forever. We've now reached the final stage, the filling of the three-chamber bags, managed by separate filling machines. We can see 3 dedicated buffer tanks, one for stream, connected to a single filling machine. These vessels function as sterile surge tanks designed to decouple formulation from filling operations, stabilize flow conditions, maintain constant inlet pressure to filling systems, and ensure uninterrupted production. In operating conditions, the final stage will consist of automated filling of the three-chamber parenteral nutrition bags. 3 chambers, 3 fillers, each filled independently. Chamber 1 will host a glucose solution, Chamber 2 an amino acid solution, and Chamber 3 a lipid emulsion. These single-chamber bags will subsequently be weighed to check the volumetric accuracy of each filler, which is a parameter that would be impossible to evaluate individually using 3-chamber bags. The 3-chamber configuration of the final product will ensure physical separation of incompatible components, preservation of critical quality attributes during shelf life, activation of final mixture only prior to administration. The Bram-Cor filling system is fully automated and designed for high dosing accuracy, sterile handling within the aseptic boundary, repeatable cycle execution, and obviously full GMP compliance. And every step remains fully traceable forever.